Welcome to this presentation on FDA requirements for peptide characterization. Peptide therapeutics demand precise scientific control, and the FDA evaluates identity, purity, structure, potency, and stability with strict expectations. In this session, we'll walk through each of these components, helping you understand what the FDA wants to see during peptide development. Peptides may appear simple, just chains of amino acids, but they are chemically fragile. They can degrade, misfold, dimerize, or oxidize faster than expected, and even small structural variations can affect potency, safety, and immunogenicity. From a regulatory perspective, the central question is, do you truly understand what you're putting into patients? Every impurity matters, and even slight structural changes can have major consequences. This is why regulators take peptide characterization extremely seriously. Understanding this is essential for successful peptide development. The FDA focuses on 5 critical areas of peptide characterization. Identity confirmation. Absolute proof that the peptide is what it claims to be. Purity and impurity profiling. Detailed evaluation of impurities and degradation products. Sequence and structural verification. Confirmation of the amino acid sequence and any structural modifications. Potency and bioactivity. Demonstration that the peptide works as intended. Stability and degradation pathways. Understanding how the peptide breaks down over time. If you can confidently address all 5 areas with strong analytical data, you're well positioned for a successful IND or NDA submission. To confirm peptide identity, the FDA expects multiple complementary analytical tools: high-resolution mass spectrometry for accurate molecular weight, MS/MS fragmentation patterns to verify the sequence, amino acid composition analysis to ensure the entire sequence is correct, retention time consistency through HPLC to support physical identity. A common mistake is relying only on molecular weight. The FDA requires sequence-level proof, not just numerical matches. Purity is essential for peptide safety and regulatory acceptance. The FDA typically expects HPLC purity of 95% or higher for therapeutic peptides. Identification of impurities present above 0.1%. Toxicological justification for any unknown impurities. Impurities may include truncated sequences, oxidized or deamidated forms, dimers, residual solvents, and other process-related contaminants. An important practical note: peptides are highly sensitive to metal surfaces. Contact with stainless steel often creates oxidation artifacts. To minimize misleading impurity data, avoid metal exposure and store samples under inert gas. Certain peptide types require advanced characterization. Cyclic peptides, where ring closure must be clearly mapped. Disulfide bonded peptides, requiring disulfide bridge mapping. Modified or post-translationally modified peptides, where modifications must be verified. Stapled peptides, where staple position and stereochemistry must be confirmed. The FDA often issues deficiency letters for incomplete ring closure mapping in cyclic peptides. Key analytical techniques include MS/MS mapping, N-terminal and C-terminal sequencing, disulfide bridge analysis, circular dichroism for secondary structure, and NMR for conformational assessment. The FDA requires functional evidence that peptides perform as intended. This includes validated potency assays that anchor batch-to-batch comparability, receptor binding studies demonstrating interaction with the intended target, cell-based bioassays measuring functional responses relevant to mechanism of action. The FDA may also expect pharmacodynamic markers and clear demonstration of the mechanism of action. A helpful industry tip: develop your potency assay early. It becomes a critical reference point throughout development. Understanding a peptide's stability profile is essential. Common degradation pathways include hydrolysis, oxidation, racemization, aggregation, deamidation, and degradation triggered by pH, temperature, or light exposure. FDA expectations include forced degradation studies, accelerated stability data, long-term stability at intended storage conditions, identification of all major degradation products. And remember, some degradants form only in solution, not in solid state. Studying both forms helps avoid gaps in your stability package. A strong CMC package must include detailed synthesis description, purification workflow, analytical methods and validation, full batch analysis data, stability reports, toxicological justification for impurities. All documentation must be consistent with clear traceability between methods and data. Mismatches, especially between impurity data and analytical methods, signal potential quality issues. Ultimately, complete and coherent documentation builds FDA confidence. In summary, peptide characterization is not just a checklist, it's a demonstration of scientific control. The FDA wants confidence that you fully understand your peptide, including its impurities, structure, potency, and stability. Consistency, reproducibility, and a deep understanding of your molecule make regulatory review much smoother. Thank you for your attention. For additional insights on peptide analytics, LC-MS workflows, or regulatory requirements, feel free to reach out.